So Sammy Weiser Novak completed his bachelor's degree in biomedical toxicology in Ontario, Canada. Sammy moved to the University of Victoria to perform his master's in the laboratory of Patrick Nearherini. Sorry if I butchered that. The author of Netter's histology. Sammy uses light microscopy and electron microscopes to study the relationship between structure and function in living things at different spatial scales. As a core facility microscopy specialist, he can help other researchers with experimental techniques that could take years and millions of dollars to develop independently. His interest in science peaked in grade 7 when his teacher showed him how to look at swamp water in his own blood under a light microscope. So he knows firsthand what a difference teachers can make. Please welcome to our virtual symposium, Sammy.
Hey, thanks so much for that wonderful introduction, Madison, and thank you all for being here today, and thanks everybody for that great talk. I think it really set the stage for this demonstration here, hopefully. So I'll go ahead and share my screen here, and hopefully there's plenty of continuity between Uri's presentation and mine. I'll try to give a little bit more of maybe a technical kind of overview over how we generate some of this data, and kind of give you a little bit of insight into the nitty-gritty. So briefly today I'll talk going from tissues to nanoscopic models and insights, nanoscopic being at the nanometer scale, and I'll be demonstrating some of this idea of getting a 3D electron microscopy data. So we'll start with some warm up with some deep thoughts, and then go on to kind of remember what resolution and means and why we bother with electron microscopy when it's expensive and takes a lot of time, and then kind of go into the technical workflow briefly and go into some of the applications that, and I'll highlight some of the work that I've had the chance to work on with collaborators, and also some of the work of my colleagues that are on the chat now at the core. And hopefully this will provide kind of a technical overview of what 3D electron microscopy can be used for, and inspire a lot of maybe more questions than answers maybe, and ideas for lesson plans and stuff, and hopefully we'll be able to work some of that out in the question time afterwards. So deep thoughts really quickly, what is life? There's an interesting paper of 2012, this idea of self-reproduction with variations. There's also this idea that inside has to be different than the outside, and that takes energy. But generally, the point is that these phenomena, how we keep the inside different to the outside, how we replicate, it all tends to happen at a very small spatial scale. Where a human hair is maybe 0.1 millimeters or 100 microns, a blood cell might be 20 times smaller than that, and a nucleus might be smaller than that, and then the other kind of constituents of cells might even be smaller than that. And the synapses that Uri was showing are much smaller, maybe 100 times smaller than a nucleus, 1,000 times smaller than a nucleus. And we'll kind of get to that in a second. Really quickly thinking about what resolution means, kind of looking up instead of down in the astronomy world, there's an idea of binary stars, and as we're looking through the atmosphere and we see twinkling stars, that's actually the water kind of getting in between our eyes and the star, and it kind of creates aberration as the light travels towards our eyes, it gets scattered, and so we see twinkling. But if we want to resolve whether a star is a single star or a binary star or a galaxy or some other phenomenon, we can correct for the aberration in the atmosphere, and so we can increase our resolution. So that resolution, the idea you can see the two spots instead of just seeing one blob. And the idea of resolving power in microscopes. So how does resolution work in microscopes? We're not just magnifying stuff, we want to be able to see not just blobs, we want to be able to resolve very small things using microscopes. And in a light microscope, you're generally limited to this idea of half the wavelength of light. To kind of put a cheat sheet up if you don't remember your light wavelengths, they correspond to color. So we're talking about 150 nanometers on a good day would be like a single pixel in a microscope image. Again, this is just to kind of emphasize that these things are on very small spatial scales, but light is limited and electrons are much smaller than the wavelength of light, and so we can kind of leverage those to electrons to get high resolution. So then there's the idea between scanning and transmission microscopes. It's really quick. So scanning, you might use a magnifying glass to look at the surface of something. Transmission, you want to cut something really thin and then you want to look through it. You want to see how light or electrons kind of interact with your sample as they pass through a thin section of your sample. So for example, you want to look at a fruit fly eye, you could, with a magnifying glass, you can see all the nice features of the compound eye, and you can start to maybe see some of the big pronounced hairs on the fruit fly, but you can't see the brain, and you can't see the guts, and you can't see the flight muscles. But if you use a compound microscope, as you see on the right, hopefully you can see those things, you can start to look at those in detail. And there's the idea between light microscopes and electron microscopes. Okay, so light microscopes on the left there, they can range in price, and you can get kind of inexpensive light microscopes, or you can get very, very expensive light microscopes that you can see on the bottom. But electron microscopes, everything has to exist inside a very high vacuum, almost a vacuum of space. And so living things generally can't survive that. And so things that we put into the electron microscopes are almost always very, very dead and also dehydrated so that they don't turn to raisins. And often heavily processed. So you can't really examine living things in the electron microscope, but you can see a much higher resolution. So here's kind of an example of like how you can use light and electron microscopes complementary ways. This is a tardigrade or a water bear that you can find in moss. You see kind of crawling around in a light microscope. You can't see all the fine features like its mouth that you might see in the bottom panel here. You can see its mouth is kind of this interesting circle shape, and here you see some of their claws in very high detail. So this is kind of how they can be complementary, but you can get different kinds of signal. So here's some more kind of data that we kind of look at in our work a little bit more. Here, using a light microscope, you can see kind of a tree in the forest of the brain, for example, and we can go into that a little bit more after, maybe in the Q&A period, if anybody's interested in talking about this organization of the brain a little bit more. But you can see like this is in an awake mouse, you can see like labeled neurons kind of pulsating, and that's how they exist in life. Here you can see in a cultured cell, you can see mitochondria, and this is from Kara Shivan that works with core and Dr. Manner. And you can see the kind of behavior of mitochondria inside a single cell. Here you can see in a behaving zebrafish, you can watch individual neurons firing as it's kind of embedded in agar, it's not moving much. So again, in light microscopes, you can get this nice resolution. You can get high resolution, you can see lots of things, you can look at dynamic behaviors, but you can't get the same resolution as inside of an electron microscope. So here we're looking at kind of what Uri showed you before, those synapses. We can start to look at those in very high detail using an electron microscope. And we can also extend those ideas to 3D and start to look at these structures that might look similar to what we see by fluorescence microscopy, but we can really investigate them in much more detail and at a different kind, and ask different kinds of questions. So I'll get back to that video later in the presentation, but hopefully that's a good kind of overview of different use cases for these different kinds of microscopes. Huh. Huh. Okay, sorry, I seem to have hit a technical challenge here. Okay, can you all still see, can you all still hear me here?
Yeah, we can hear you. And I can see your, see like your whole PowerPoint window.
Oh, perfect. Okay, sorry, yeah, just kind of froze out on me there. So now we can just jump right into the technical workflow. So the idea is if somebody comes to the core with a sample that they want to look at with using 3D electron microscopy, this is kind of what the workflow might look like. We receive the samples, we fix them with aldehydes so that they don't rot or anything like that. So they're very preserved with very high structural integrity. Then we can stain them with heavy metals. We can embed them in plastic. We can section them with diamond knives. We can image them with these electron microscopes under high vacuum. And then we can, the rest of this will become a little bit more, make a little bit more sense as we move through the workflow here. So briefly starting with the dissection idea, this is Leo Andrade, the electron microscopy master that works with the core. And here he's dissecting the inner ear, kind of what Uri was showing in his presentation. This is what the first step looks like. So if we were to get an inner ear from a collaborator, we have to go through, or Leo has to go ahead and dissect away all of the fat and bone and things that aren't the tissue that we want to look at. This can take an hour or for a single ear to dissect. And here you're starting to see him to pull out the spiral cochlea there. And then he has to take off the tectoral membrane, which is what those hair cells embed themselves in that Uri was talking about. And so this is a very challenging preparation, all under a microscope with very steady hands. And then after you dissect the sample, you fix it with aldehydes, and you stain with heavy metals like osmium and uranium that can interact differently with the electron beam, and they bind to things like the cell membrane and to different proteins. So we can provide stain to provide gain for the features that we want to look at in our sample. And then we can further process it by replacing all the water with ethanol, and then replacing all the ethanol with an epoxy resin, curing that in an oven, and then we can prepare that. And this is what that cochlea might look like. This piece, that spiral, looks like inside of a piece of epoxy resin. So this is a little, after it's stained with metals, it kind of turns black. It's this little black strip. And so this is what that can look like under a scanning electron microscope. This is what Uri showed in his presentation. But if we want to look at it, if we want to look at the inside of these cells, you can't see that with the scanning electron microscope in quite the same way. So what we can do is we can cut serial sections. So on the left here, you can see, this is called an ultramicrotome, which is kind of like a really, really fancy deli slicer. And what it does, we put a diamond knife on it. So this is a perfect diamond edge, about 6 millimeters wide. And we have the sample kind of advancing very slowly, about 70 nanometers every time you want to take a slice. And we have it's called a boat, but we have the knife filled with water. So that as the sections come off of the knife edge, they float out on water and they create this perfect ribbon. And then we can collect those ribbons on silicon chips that you can see here. Some of them are more perfect than others. And then we can put these chips inside of the electron microscope. And so once we look at them inside of the electron microscope, we can start to see some of the detail. And hopefully this starts to look familiar based on the video that Uri showed in his presentation as the inner ear cochlea and cross section, right? So here we can see that there's axons that will go to the brain to transmit the signal from the hair cells up here in this box. So the hair cells will synapse with neurons that project from the brain. They'll go all the way back to the brain and transmit that sound signal to the brain so that we can interpret it and make sense of it. And we can see here blood vessels with blood cells in them that are supplying energy. You can see the bone here stained in black. And we can see the myelinated axons. This is almost like you have if you were to have wires, you always keep wires insulated, because you don't want to lose signal as they travel through your house or through whatever project you're making. So myelin acts as insulation in the body. So you can have long-range projections of neurons, and they don't lose signal as they move sometimes centimeters or even all the way down your leg. There's just one neuron called the sciatic nerve that can be up to 1 meter long. And so myelin is really important in that case to make sure that you can feel your toes well and you don't get chronic pain and things like that. So, but if we look at these hair cells a little bit more closely, the cells that kind of transmit the sound, we can kind of get a sense of the difference between what we can see with the scanning electron microscope versus with the transmission electron microscope, or by serial sectioning the electron microscope. And the idea is we can see the inside. So here, if we look in this square, you can see that there's a nucleus here. This is where the DNA is. And we can highlight all of the mitochondria here. And we can look at one in particular, and this is where all the energy is made for this, or well, where energy is made and kind of converted into usable energy for the cell. And so we can, and as Uri demonstrated in his presentation, the mitochondria can respond to injury and they can respond to all kinds of conditions. And so understanding the distribution of mitochondria in a cell might be a really important question to ask. And so how do we start to get 3D information from these serial sections? Well, we can stack them all up and we can align those sections. And once we do that, we can wind up with something like a block here of tissue. And if we segment, kind of color in the lines for each mitochondrion and for the nucleus, we can start to get some 3D representations of these individual components of these cell types, right? And that's how we can start to build these 3D models. And then from there, we can go towards analysis. And I won't go too much into analysis today. I'm going to kind of stick more to visualizations. This is from Tammy Pham, who's a medical student who really was a wonderful collaborator, and she kind of did a lot of this tracing work and also did this visualization, which I think is just wonderful. So, and she's kind of leading the analysis side of this project in terms of trying to figure out how mitochondria are distributed differently before and after noise. So this is kind of how we can start to study that. You can imagine you can just take a distance from the nucleus for each mitochondrion, the center of each mitochondrion to the center of the nucleus, and you can look at the different distributions of distances based on before and after noise and start to understand if that's really a salient response to that injury there. So for the next part of the talk, I'd like to highlight some of the work that I've been, that myself and other collaborators in the core, or other my peers in the core, Leo Andrade, Linjing Fang, and Arlo Sheridan, have been working on over the last few years, and kind of projects that we're working on day to day here. So here's a project that we worked on in terms of where we were looking at the brain, and just a refresher on how the brain's organized. Brain cells are kind of organized like trees, where you have branches that receive signal, kind of like how leaves receive light, or you have leaves on branches, how that receive light, and similarly you have these dendritic spines with synapses on dendrites that receive signal in the form of neurotransmitter, kind of how Uri explained earlier. And here you can see that these synapses can happen on dendritic spines, which is what we're looking at in this right now in the video here. You can get a sense that the blue is the spine here, and it kind of plugs on to this axon here. And here we can also see that sometimes synapses happen on the shaft of the dendrite, where here we see that there's some mitochondrion and a bunch of synaptic vesicles right opposed to this dendrite here that also has some mitochondria inside of it. And the relationship between shaft synapses and spine synapses is really important for information processing in the brain. And so that's, and this is really one of the most effective ways to study these kinds of phenomena. So, but it's very time consuming to trace out these processes by hand. So kind of what Uri hinted at, and what Arlo Sheridan is really an expert at in the core, is how do we take those human training data sets and extend those just to massive data sets? For example, an entire fruit fly brain. And so here's an example of what the training output can look like. So here is Arlo's program filling in a single dendrite, how I might try to do it, but it might take me 100 hours to do this single dendrite really precisely, where it might take Arlo's algorithm on a really, really powerful computer, it's mere minutes. And so you can imagine how this can scale to a really dense volume. If you want to segment all of the features in here, it would take a very hard to quantify amount of time for a human to segment this whole volume in such a saturated fashion. In fact, one of the leaders in the field, Kristen Harris, has been building a saturated volume by hand for almost 20 years, and she's still kind of updating it with detail to this day. But you can see that Arlo's algorithm can do this in, again, matter of minutes or hours automatically using the training data sets, and this can be extended to the whole brain, where we can determine in a fruit fly different projections from one region to another, and even calculate all the different synapses between cells from one region that project into another region. And so I encourage you to ask Arlo about some questions about how he did that in the chat. But as kind of already mentioned, it would be totally impossible. A fruit fly brain is one thing, but a mouse brain would have, or a human brain would have more information than the whole internet. And so it's not really practical to image at very high resolution. If we want to count all the vesicles in every synapse, we simply couldn't do that at this point. So an idea that what we could do instead is we could image everything at low resolution, and then image subregions at high resolution, and train a deep learning net to be able to calculate what the high resolution looks like from the low resolution. And that's exactly what Linjing did here. And that enables us to do very very effective segmentation, where we can look at the relationship between not just one cell and another cell, but inside of one cell, looking at different components of the cell, like vesicles and endoplasmic reticulum and mitochondria, and seeing how those relationships change under different conditions as well. And then changing gears completely, happy Halloween, everybody. This is another idea of what we can look at inside of a scanning electron microscope. So this is an image that Leo Andrade took of a spider. I'm sure most of you would recognize it, but maybe you didn't realize just how hairy spiders can be. And maybe you thought, like, well, you know that spiders have mandibles, you know, they have eyes, but maybe you've never realized that they had so much hair around their eyes. And maybe we could take a closer look at, you know, what that hair around their eyes looked like. And you can start to see how there's different lengths of hairs. And if we go a little bit closer, you can see that some hairs even have lots of bristles. And you can see that there's actually stomata, and this is how air exchange can happen in insects. They don't have lungs like we do, so they have little holes all over their body where they can achieve air exchange. So this is another example of kind of how the scanning electron microscope can kind of give you a different kind of insight into everyday things. And with that, I don't want to go over time. I do have a few more slides here, but I wonder if it's time for a Q&A at this point. So I'd like to thank everybody at the core and you all for your attention, and thanks so much for the opportunity to present some of our work here today.
Awesome. Yeah, we have a couple minutes for Q&A. If anyone, you're welcome to unmute yourself if you want to ask verbally, or continue to type them into the chat. I did have one quick question. Why does this, in one of the early slides, you showed and said that it needed to be in a vacuum? Why does it need to be in a vacuum?
Yeah, so an electron beam is kind of different to a light beam in that electrons can interact with air and other kinds of particles in atmosphere. So you can't just shoot an electron beam; it'll interact with everything. You can't, and so you have to make sure that there's no particles in your chamber where you want where you're focusing your electron beam. And so you need to pump down that chamber down to almost the vacuum of space.
Awesome, thank you.
That said, there is a group in Japan that has a really interesting technique to create almost like a body armor that seems to work for some insects. You can look at them in a scanning electron microscope while they're behaving. But it's not a very popular technique yet because it's quite new, but it's pretty exciting.
Looks like we do have a question in the chat from George. Are quantum computer architectures influencing brain function hypotheses?
That is a great question. Maybe somebody in the chat can field that better than me. I'm not a quantum computing guy. I think that if you wanted to look at, if you're building a quantum computer and you wanted to make sure that you have like quality control or something, or for all computing applications, really like quality control is done inside scanning electron microscopes, and most processors that you're using in your computers to watch this video are built in focused ion beam milling machines, which operate under vacuum. And we use those also for some of the imaging that I presented. We use focused ion beams combined with scanning electron microscopes so that we can mill away one layer and then image, and then mill away the next layer and image inside of the microscope. And that way we can also get much higher spatial resolution in Z. But that's not a scope that we have at Salk right now, but it's one that I've worked on before in Canada. So we have access to some microscopes like that. And we have collaborators that work with microscopes.
Awesome. We have a question from both Robert and Adrian. Would like to know, does your work require coding and biological degrees?
Oh, that's a great question. So I'll say my degrees are in biology. Firmly, and so Arlo can really testify to my I'm filling holes constantly. But I think every time when I've had the opportunity to speak to students in high school or middle school, generally if they're thinking about college applications and if they're interested in biology, I generally recommend they go into something like physics or engineering for an undergraduate degree, because generally speaking, I think biology from my perspective, looking backwards, it's a lot easier to just teach myself some of the biological concepts than to teach myself linear algebra or differential equations, or how to apply those in those kinds of ideas in a programming framework. That said, there are lots of great online courses through Coursera. So I taught myself Python and a bunch of other programming languages online, just in order to do the tasks that I needed to do at the time. And then over time become more or less proficient in being able to cobble together tools. But I think if I had a foundation in computer science or engineering or physics, where those kinds of kind of more technical skills are more enforced, or more like kind of part of the curriculum, I guess, that might, looking back, that would be really helpful for me right now. Yeah. Hope that answers the question.
I want to tell a story about one of my heroes in the imaging field. His name was Michael Davidson. He died of cancer a few years ago. But one of the amazing things he did, so first of all, building on what Sammy said about degrees, it's much easier. If you have a degree, it opens doors. There are certain grants you can get, certain types of jobs you can get if you have a PhD or a college degree that you can't get without. But there is one guy named Michael Davidson who decided to buck all of the conventional trends And he, first of all, got private funding by selling microscope images. So he built his entire research operation through sales and decided he didn't care about the government, didn't want funding from any of the traditional sources. And the other thing he did that was really cool was he hired high school students and dropouts. He had a soft spot for kids who had suffered or made mistakes or something in the past that made it harder for them to get degrees and to go down traditional paths. So he would hire these high school students who his only requirement was that they were smart and motivated. And he taught them biology, how to do molecular biology, how to do microscopy. And there are a lot of examples of that with computer coding as well. These things seem esoteric because they're not part of our daily lingo. But the reality is that everything can be broken down into simple components and anyone can learn them. And you shouldn't be daunted by whether you have a degree or not. It's within your grasp. It 100% is. So that's my answer to that question. I hope it at least provides some sort of answer.
Thank you. Looks like we have time for probably one more question. If anyone has any final questions for our presenters today. Again, you're welcome to unmute yourself if you'd like to ask verbally.
Or maybe I'll just give another plug for there's the software that I use for all the 3D stuff. I try to use all free software, and so it's all open source free software for all the analysis. And a really cool piece of software that I've been working with for a while now is called Blender 3D, and that and there's some really wonderful tutorials. You can make full-length animated movies in it, like similar to Pixar movies. There have been a number of movies made in the last few years to kind of show off all the different kind of workflows and kind of tools that you have built into the software. And it's really one of the most exciting things that I'm working on these days, and it's totally accessible for students. And there's a really wonderful YouTube channel called Captain Disillusion, where he goes over a lot of the, he uses Blender for a lot of stuff. He also talks a lot about a lot of these concepts like resolution and frame rate and color, in really, really cool, intuitive ways. And I think that they could be integrated into lesson plans well. And I can kind of pop those in the chat now as well. That was something I was thinking about this morning as something to plug. So maybe we'll do that at the same time here.
All right. I would like to say thank you so much to the Biophotonics Core team for joining us today. Your passion was evident. It definitely came through, and you definitely sparked a lot of ideas for myself, even including it for myself, and ways that we can show our students that while science is important, understanding the tools that we use to do science is just as important. So I'd like to say thank you so very much for joining us and spending a couple hours with us.